Welcome to another episode from the Veterinary Research Channel. Today, we're diving deep into a fascinating study that explores the relationship between Giardia duodenalis, a common intestinal parasite, and the gut microbiome in dogs. Does the microbiome play a role in whether a dog develops diarrhea when infected with Giardia? Let's find out. Giardia duodenalis, or GD, is a protozoan parasite that infects various animals and humans, causing giardiasis. It is considered the most common cause of parasitic diarrheal disease in dogs globally. The clinical signs of GD infection can vary significantly. Some dogs remain asymptomatic carriers, while others develop severe diarrhea and malabsorption, particularly young or immunosuppressed animals. Giardiasis also has zoonotic potential, highlighting its public health significance. The clinical relevance of giardiasis is often debated because many infected individuals do not show clinical signs. However, some hosts develop diarrheal giardiasis, which can become chronic. In humans, post-infectious syndromes like irritable bowel syndrome and food allergies can occur even after the parasite is no longer detectable. Additionally, Giardia's resistance to common treatments such as fenbendazole and metronidazole is increasing, making it crucial to investigate virulence, resistance mechanisms, host immune responses, and alternative treatments. The intestinal barrier, which includes the intestinal microbiome, mucus, and epithelial lining, is disrupted by Giardia, initiating the pathophysiology of giardiasis. Virulence factors like cysteine proteases contribute to the severity of the infection by disrupting intestinal epithelial junctions, depleting mucin, degrading host immune factors, and breaking down microbiota biofilms. Host and environmental factors, such as immune status, age, environmental stress, and concurrent infections, also influence the disease's outcome. The interaction between Giardia and the gut microbiota has become a focal point in understanding disease pathogenesis and potential therapeutic interventions. The commensal microbiota can control the colonization and establishment of Giardia in the gut. Studies in mice have shown that protective anti-Giardia microbiota can be transferred to prevent Giardia establishment. Furthermore, dysbiosis, or an imbalance in the gut microbiota, contributes to clinical signs associated with giardiasis in both mice and humans. Dogs naturally infected with Giardia have been observed to have increased gut microbiota diversity. However, previous research in dogs has been limited by small sample sizes and heterogeneous cohorts, making it difficult to draw firm conclusions about the role of the intestinal microbiota in the severity of giardiasis. To address these gaps, a recent study aimed to investigate the associations between fecal microbiome characteristics and gastrointestinal clinical signs in dogs naturally infected with Giardia. The study focused on a relatively large and homogeneous group of dogs, assessing the fecal microbiome features in those with and without diarrhea. The study included fecal samples from kennel dogs diagnosed with Giardia using a fecal Giardia antigen, ELISA. The researchers compared the fecal microbial composition and dysbiosis index, DI, between dogs with diarrhea and those without. The study design was cross-sectional, spanning from 2019 to 2021, and included dogs from municipal shelters, private shelters, and a commercial breeder kennel. All dogs were fed commercial dry diets. Dogs younger than 4 months or older than 5 years were excluded to minimize age-related variations in the fecal microbiome. Shelter dogs were included only if they had lived in the shelter for at least 3 months to ensure sufficient historical data collection. Information on the dog's general health, defecation frequency, and fecal consistency was obtained from kennel staff. Dogs were included only if they were otherwise healthy, based on their history, attitude, appetite, and physical examination. Dogs treated with nutritional interventions or drugs affecting Giardia or the fecal microbiome, such as antibiotics or proton pump inhibitors, within 3 months before fecal sample collection were excluded. Fecal samples were collected from individual kennel cages or during walks and tested for Giardia infection within 1 hour of collection using the Giardia antigen, ELISA. Positive samples were immediately frozen at -80°C for later microbiome analysis. Fecal consistency was visually assessed upon collection using the Purina 7-point fecal scoring chart, with diarrhea defined as a score of 4 or higher. DNA was extracted from the fecal samples using a Magmax microbiome ultrakit, and the V4 region of the 16S rRNA gene was amplified using PCR. The resulting amplicons were purified, quantified, and sequenced using the Illumina MiSeq platform. Microbial communities were analyzed using KIME2, and taxonomy was assigned against the Green Genes database. Alpha diversity, which measures fecal microbial richness, and beta diversity, which measures microbial community composition overlap, were compared between the diarrheic and non-diarrheic groups, as well as by age and sex. Analysis of microbiome composition, ANCOM, was used to identify differentially abundant taxa between groups. The Canine Microbial Dysbiosis Index, DI, was determined by at fecal samples to the gastrointestinal laboratory at Texas A&M University. The DI uses mathematical modeling based on specific fecal bacterial abundances relative to a reference set from healthy dogs. The resulting numerical value is interpreted as normal, 0, mild to moderate shift, 0 to 2, or clinically relevant microbiota shift, 2. Statistical analyses included the Kolmogorov-Smirnov test to examine data distribution, Fisher's exact test to compare qualitative variables, and the Mann-Whitney test to compare quantitative variables. A p-value of less than or equal to 0.05 was considered significant. The study collected fecal samples from 131 dogs residing in various shelters and a commercial breeder kennel. Giardia duodenalis infection was diagnosed in 42 dogs, 32%, with 23, 55%, experiencing diarrhea and 19, 45%, being non-diarrheic. All diarrheic dogs showed clinical signs consistent with small bowel diarrhea, such as infrequent, large volumes of soft to unformed feces without blood or mucus. After DNA extraction, 38 samples were available for 16S rRNA gene sequencing, and 19 samples from each group were available for DI analysis. The researchers found no significant difference in Faith's phylogenetic diversity, PD, between diarrheic and non-diarrheic dogs. However, when divided by sex, diarrheic males had higher alpha diversity compared to diarrheic females. No age-specific effects were identified. Beta diversity analysis, based on weighted unifrac distances, revealed no significant differences between diarrheic and non-diarrheic dogs overall, or when examining sexes and age classes separately. Similar results were obtained using different metrics such as unweighted unifrac, Bray-Curtis, and Jacket. When examining specific taxa abundances, no significant differences were found at the phylum, genus, and species levels between diarrheic and non-diarrheic dogs overall. However, the genus Clostridium and the species Clostridium spiriform were more abundant in fecal samples of male diarrheic dogs compared to male non-diarrheic dogs. Proteobacteria were more prevalent in fecal samples of diarrheic dogs younger than 1 year, while varicomicrobia were more prevalent in older diarrheic dogs. The overall fecal DI did not differ significantly between diarrheic and non-diarrheic dogs. The quantities of the 7 specific bacterial taxa constituents of the DI were also similar between the two groups. This study examined the associations between fecal microbiome characteristics and the occurrence of clinical giardiasis in a cohort of young, healthy dogs infected with Giardia, housed in similar kennel environments, fed dry commercial diets, and not exposed to previous nutritional or pharmaceutical interventions. Contrary to previous studies in humans and animals, which reported significant intestinal microbiota composition shifts in Giardia-infected hosts, this study suggests that fecal microbiome alterations do not differentiate diarrheic from non-diarrheic dogs. The results indicate that host and Giardia-related characteristics, such as immunocompetence and parasite virulence, may play more significant roles in the clinical expression and outcome of giardiasis in dogs, while the intestinal microbiome plays a more minor role. Several factors may explain the differences between this study and previous studies regarding the impact of microbial dysbiosis on the clinical signs of giardiasis. Previous studies often used in vitro or experimental murine giardiasis models, which may not accurately mimic the natural infection characteristics in the current cohort. For example, acute experimental infections with Trichomonas fetus in cats result in profound dysbiosis, whereas naturally infected chronic carriers do not. Chronic T. fetus infection may even confer beneficial microbial changes, such as increased abundance of short-chain fatty acid producers that support colonocyte health. Similarly, previous studies have reported Giardia-induced microbial changes, including increased fecal microbial diversities and increased beneficial microbe abundances. These changes may persist regardless of diarrheic status. Additionally, previous studies of natural giardiasis in dogs often included small cohorts that were heterogeneous in age, diet, past drug treatment, and habitat, introducing potential confounders affecting fecal microbiome composition. Young age is a known factor determining the clinical severity of giardiasis in dogs, likely due to immune system immaturity and higher co-infection prevalence. Age also affects the microbiome, influencing its interplay with parasitic infections. Chronic subclinical giardiasis in puppies is associated with dysbiosis and increased fecal calprotectin concentration, indicating chronic low-grade inflammation. These changes may not be associated with clinical disease, but could explain microbial feature differences among studies with heterogeneous ages. In this study, proteobacteria were more abundant in young diarrheic dogs compared to older diarrheic dogs. Proteobacteria are dominant in the gut of young mammals and play a crucial role in preparing the gut for colonization by strict anaerobes needed for healthy gut function. Comparing microbiomes and their clinical associations in dogs of variable ages with giardiasis may skew results. In this study, the group signalments were similar, and the main fecal microbial diversity parameters did not differ between diarrheic and non-diarrheic dogs, even when assessing age and sex groups separately, eliminating these signalment differences as potential confounders. Clostridium spp, particularly Clostridium spiriform, were significantly more abundant in diarrheic males compared to non-diarrheic males. Sex-associated differences in gut microbiota composition may play a role in sex differences observed in various diseases. Therefore, the association of Clostridium spp with diarrheic status in male dogs with giardiasis cannot be excluded. The lack of associations between fecal microbial composition and the presence or absence of diarrhea in Giardia-infected dogs is further supported by the similar DI of both dog groups. The fecal DI of most dogs in this study was a 0, suggesting normobiosis. Although dysbiosis and abnormal DIs might be expected in dogs with diarrhea, profound dysbiosis was absent in the majority of diarrheic dogs. The presence of diarrhea does not necessarily indicate dysbiosis, as demonstrated by DIs within the reference range in various acute and chronic gastrointestinal diseases of dogs. The DI recorded in this study suggests that dogs with giardiasis do not suffer prominent dysbiosis, and dysbiosis is not the primary contributor to diarrheal disease development. Several limitations should be considered when interpreting the findings of this study. The cohort size may have negatively affected the power of the statistical analyses, particularly regarding female group proportions, Faith's PD, and DI of diarrheic dogs. However, microbiome analyses showed no differences in sex between diarrheic and non-diarrheic dogs, and weighted unifrac beta diversity results support the conclusions. Although investigating kennel dogs decreases habitat heterogeneity, this design feature may have been associated with inferior monitoring compared to privately owned dogs, as clinical signs were assessed based on a single visit. Nevertheless, historical information from experienced kennel personnel who monitored the dogs several times daily allowed the classification of diarrheic and non-diarrheic dogs. Thorough historical and physical examinations by kennel medical staff and investigators upon fecal sample collection further minimize the chances of erroneous health and fecal consistency assessments. The diets were not standardized, which may have affected the clinical signs and fecal microbial composition. However, all dogs were fed dry commercial diets exclusively, with identical ingredients and similar nutritional profiles, minimizing potential effects of diet types on the results. A control kennel dog group uninfected by Giardia was not included. However, examining the DI allows for the assessment of fecal microbiome deviations compared with the reference interval, compensating for this limitation. A recent study comparing the fecal microbiome of asymptomatic Giardia-infected dogs to healthy dogs concluded that Giardia presence is associated with the enrichment of protective bacterial taxa, which may limit host inflammation and cause only minimal modification of gut microbial ecology. Thus, Giardia-infected dogs do not sustain significant microbial compositional shifts compared to healthy dogs. Giardia infection was diagnosed by fecal Giardia antigen detection, not by molecular testing, precluding the identification of specific Giardia assemblages. However, diarrhea presence has been reported to be unassociated with particular Giardia assemblages in dogs. The study did not investigate the presence of parasitic co-infections, precluding the examination of their potential impact on clinical signs and the fecal microbiome. Dogs with giardiasis show different microbial composition compared to healthy dogs, but no such difference is found between Ancylostoma caninum positive and negative dogs. Exclusion of hookworm-positive dogs from Giardia-infected dog studies strengthens the differences between Giardia-positive and negative dogs. This suggests that concurrent parasitic infections somewhat affect the extent of giardiasis-associated intestinal microbial changes. However, Ancylostoma caninum infestation is absent in the region where the study was conducted, and extensive deworming protocols were implemented routinely in all kennels, decreasing the chances of clinically relevant helminthic co-infection and any impact of such co-infection on the results. In conclusion, while humans and dogs infected with Giardia show intestinal microbial compositional changes compared to non-infected individuals. This study's results suggest that fecal microbial composition does not differentiate symptomatic from non-symptomatic dogs with giardiasis. Profound dysbiosis, as reflected by the DI, was not a common feature in either group of Giardia-infected dogs. These findings suggest that other host and parasite characteristics play important roles in determining the severity of giardiasis in dogs and may affect the host microbiome interplay in Giardia infection. Given their shared environments, similar omnivorous diets, and intestinal structure, dogs serve as an ideal model system for translational gut microbiome research in humans. Because previous studies in mice suggest that microbiome manipulation using pre- and postbiotics may benefit the management of Giardia-infected hosts, this study's findings suggest that such an effect may not translate into dogs with natural Giardia infection, warranting dedicated in vivo studies of dogs and humans. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.